Hello everyone, good day. I am Apnilin O. Escopete from Central Philippine State University San Carlos Campus, Negros Occidental. I am one of your presenter and Professional Education 1 from BSED MATH-2B. My study is all about a lifespan perspective on brain development, longitudinal studies of gray and white matter maturation. Our brains are constantly changing throughout life with gray and white matter developing in gray shaping from infancy all the way into old age. By following the same people over time, longitudinal study give us a clearer picture of how the brain truly grows and adapts, rather than just comparing different age groups. Gray matter tends to grow quickly in early life and then just refine throughout pruning while white matter slowly strengthens connections well into adulthood before gradually declining. Understanding these patterns helps us recognize what healthy brain development looks like, spot early signs of problems, and learn how our brains support thinking and memory across the lifespan. Longitudinal development of human brain wiring continues from childhood into adulthood. This study only covers ages 5 to 32, missing key developmental periods such as late adulthood and aging. As a result, conclusions about whether white matter maturation continues or declines into later life can't be drawn. Structural brain development between childhood and adulthood convergence across four longitudinal samples. The analysis does not link structural changes to behavioral, cognitive, or functional outcomes, so the significance of the volume trajectories for real world development remain unclear. Longitudinal development of human brain wiring continues from childhood into adulthood. To track how white matter pathways in the human brain development and strengthen from childhood through early adulthood. Structural brain development between childhood and adulthood convergent across four longitudinal samples. To map how the brain structures develops from childhood to adulthood by comparing patterns across four longitudinal studies. Who participated? 103 healthy children and young adults ages 5 to 32 for white matter analysis and 391 participants ages 8 to 30 across four studies for brain structure analysis. How the brain was studied. DTI or diffusion tensor imaging to look at the fine wiring of the brain's white matter. MRI to measure the size of gray and white matter as well as overall brain volume. Analysis. Track changes in each participant over time and look at how growth patterns differed across brain regions and between male and females. My finding is white matter tracks develop in different ways. Some mature by late adolescence or projection in commissural tracks, while others continue developing into early adulthood, association tracks. Changes in white matter are mostly due to improved organization and insulation of brain fibers, not just bigger size. Gray matter peaks in childhood and gradually decreases during adolescence, while white matter keeps growing until mid to late adolescence. Taking brain size and sex into account help us explain differences in regional brain development.
Hello, good day everyone. I am Ranel Pre Cargente from Central Philippine State University San Carlos Campus, Negros Occidental. I am the second presenter of Professional Education 1 from BSED MATH-2B. So let's proceed to the conclusions. Brain development is a long, ongoing process. Gray matters grow early and decline in adolescence, while white matters continues to refine well into young adulthood. This prolongs develop, especially in the brain. Brain connections may help explain the growth of complex cognitive skills during adolescence and early adulthood. So the insight of our study is similarities and differences. So label et al. 2011 focused on white matter development or track specific, and method is longitude DDI. Findings, white matters matures into adulthood. Sensory motors track faster than frontal association track. So while Mills et al. 2016 is he focused gray and white matters, multiple samples convergence. So his methods is for longitude MRI datasets. So findings: gray matters decrease, white matters increase. So region specific consistent pattern. So another insight is reflections and implications. So brain development continues into adulthood, supporting ongoing learnings, executive findings, and cognitive skills matured late. So impact education strategies and regional differences. So regional differences as personalized learning approach. And the last label is detained neutral wirings, Mills generalizable development trends. And the last one is the recommendation. So track brains develop over time with longitudinal studies to understand both structures and functions. Next, special attention to association track that matures later, as they are linked to complex thinking and behavior. Next, include individual differences like brain size, sex, and genetics into future studies to better understanding development. And the last one is use these findings to inform early interventions for neurodevelopmental disorder. So that's all for today. Thank you guys.